The brain disease chronic traumatic encephalopathy, CTE, may stem from rampant inflammation and DNA damage, according to a new study. The research reveals that head impacts can trigger inflammation and DNA damage in brain cells, leading to cell dysfunction and death over time. This DNA damage is similar to that seen in Alzheimer's disease, suggesting a potential link between head trauma and neurodegenerative disorders. Scientists behind the study discovered that mature neurons, which don't divide, accumulate mutations throughout life. These mutations build up faster in brain diseases like Alzheimer's. Dr. Christopher Walsh, a geneticist, explains that neurons pick up mutations year after year, and these mutations accelerate in neurodegenerative diseases. This finding raises the question, could DNA damage in other brain disorders also drive neuron loss in CTE? The study analyzed the genomes of individual neurons from people with and without CTE using single-cell whole genome sequencing. Results showed that neurons from CTE brains carried more DNA mutations than healthy brains, with about 114 additional single-letter changes per neuron. Neurons from individuals with repeated head impacts but no CTE showed no increase in mutations. The pattern of mutations in CTE resembles Alzheimer's disease, with both conditions showing increased mutations and similar DNA alterations. Another type of genetic damage, short insertions and deletions, indels, was also identified. These tiny DNA breaks were more abundant in neurons from CTE and Alzheimer's brains, with some CTE cases containing over 1,000 indels, equivalent to a century of normal aging. Dr. Walsh suggests that these indels could cause serious dysfunction or death in affected cells. While the study didn't directly test for inflammation, earlier research by co-authors Dr. and McKee and John Cherry has shown widespread microglia activation in CTE brains, indicating inflammation. The team believes CTE might result from repeated head trauma and inflammation, bombarding the genome with damaging processes similar to UV light or tobacco smoke. This combination could lead to DNA damage and neuron loss. The study's findings highlight the role of inflammation in CTE, suggesting that head trauma triggers inflammation, which promotes DNA mutations and cell dysfunction. The researchers are now investigating whether similar processes occur in other neurodegenerative diseases, aiming to understand the biochemical steps from inflammation to neuron death and potential intervention points.